Thank you, everyone. That was a wonderful series of thought-provoking talks about the human brain. And on behalf of CARTA and my co-chair, I'd like to start the Q&A session. So speakers, if you haven't already, please turn on your cameras, unmute your microphones. And we've collected audience questions throughout the day, and we will address them now. So with that, I'm going to present the first question, and this is addressed to Dean Falk. Does the theory you posed, linking auditory entrainment and language to a mother's footsteps, rely on the assumption that language first evolved from vocalizations? Some researchers argue that our linguistic capabilities evolved first in gestures and later in spoken language. So I'm wondering if that view could also be consistent with your hypothesis.
We do address the vocal channel. Mainly, we're well aware that gestures are part of the package. And in fact, when you talk about Mother East, for instance, we talk about baby talk and the vocalizations and the affect and the prosody, but also there's a huge component that is gestural. We acknowledge that, but we did not speak to that or incorporate a theory to do with the evolution of gestures per se within the ideas about entrainment of movement to sounds. And thank you for the question.
So do you know the time range for novel one selection sweep? An environmental disturbance relating to lead makes me think of volcanic trends. As volcano plumes deposit large amounts of lead and other heavy metals like cadmium, and contemporary people who lived through them exhibit elevated urinary and blood concentration of these heavy metals for long periods afterwards. That's a great question. It's hard to estimate when we see the nova ones swap, but we estimate about 200,000 years ago. So it's quite recent. But definitely the environmental exposure was there before that. So the genetic comes later.
Great. I have the next question for Nanad Seston. Nanad, the person asks, I'm curious about the role of retinoic acid in shaping the neurodevelopment. In immunology, retinoic acid guides T cells to go to the gut and also regulate immune cell development. I'm wondering if some of retinoic acid's effect on brain development could actually be indirect, working through the immune cells it shapes rather than acting on neurons themselves.
Thank you. That's a very good question that I don't have a direct experimental answer. Just as try to a little bit speculate, I would not be surprised that that brain does not develop in a vacuum. So I would not be surprised that immune cells don't have effect. We can see macrophages that come from immune cells and microglia that is a part of immune cells. So that's not native to the central nervous system as early as even early fetal to early mid-gestation in humans and other species. So that would not be surprising. We don't have any evidence that these cells do release. The source of retinoic acid seems to be multiple cell types, not just neurons. Also, we see glial cells. We see cells outside the cortex too. Also, it's a very promiscuous signaling pathway that affects many cells and systems, not just within the brain. So I would not be surprised at that effect. But we don't have any evidence for it. But we cannot exclude that. Thank you.
So I have one that is for me as well, from Adrian. In the Nova 1 Neanderthalized organoid experiment, were those homozygote genomes introduced or heterozygotes? And if the later case, which gene is dominant? Curious how the Neanderthal sapiens hybrid phenotypes would have fared with the potential lead exposure. For the lead experiment, we only work with homozygotes. So these are clones that have either the two copies of the modern version or the two copies of the archaic version. So we haven't done this experiment that you're suggesting, which is pretty cool.
I have one for Joe Paradiso. The question asks, in your project, jam_bot, you describe the AI model as acting like a sort of musical brain. Based on your research, how does this musical brain behave differently than the human composer's brain? How do you see the collaboration between the two evolving?
We've, at least working with living composers like Jordan Rudess, the person we've worked with most closely, he provided hours of data of him playing in different styles, and basically it took a transformer model that was developed for generic music and honed it into his playing style, so he could kind of anticipate what it was going to do, because it's kind of what he would do. In the beginning, especially, it would take some wild jumps once in a while. You see Jordan kind of smile and laugh and play another line to kind of bring it back. So he called it Baby Jordan. Once in a while, it would get kind of frantic and take a tantrum, so to speak, and he'd bring it back. As we honed it a little more with reinforcement learning, it got a little better behaved. So it's more laminar, and it still would give him ideas, but they'd be within the bounds of kind of what he would expect. So it is kind of a partner where you can trade lines back. For me, playing with Jordan's model, I would never play those chords because I don't know them. He does. So I have somebody else's expertise at my disposal that's looking at what I'm doing to come in and bring me to a different place. So yeah, certainly as you train the model, it evolves in its capability. They're just going to keep getting better. But the same token, as a player working with it, it can put you into new places. So it works on both ends.
I have one for Bruce Miller. Question is, or the statement, you explain in your talk that as patients begin to experience the symptoms of SVPPA, they often develop increased visual attention and artistic output, which you argue reflects something evolutionarily fundamental about the artistic nature of the human brain. Do you think there could be any shared neural mechanisms between the visually heightened experience associated with SBPPA and those seen in synesthesia?
With Ann Adams in particular, we wondered whether she had been synesthetic because she was really presenting individual notes visually. And, looking at her notebooks, I think this may have been a component of her creativity, that, a transmodal association of sound with a visual output. So I think it's a really smart question. And I think in some cases that really does occur. Yes.
There was a question to me asking, I mentioned evidence that OPCs can participate in synaptic pruning. Do you think this contributes to the increased cortical plasticity seen compared to other primates? Yeah, that's a great question. So the evidence for OPCs contributing to synaptic pruning is actually found in rodents. So it's not something specific to primates or specific to humans. But I tried to lay the stage or propose that if we have more OPCs, perhaps there's more around to do this in a more efficient way, or to participate more broadly in this compared to other species. So it's something that we're trying to test in a dish by doing these mix and match experiments of human and chimpanzee OPCs with neurons to test, at least between those two genetically closely related species, whether there's a difference in activity or function. But yeah, it's really speculative at this point. So great question. Sorry that I'm doing all the talking, but I'll just continue asking the questions. For the next one's for Miles Wilkinson. The HAR-123 knockout mice showed a decrease in cognitive flexibility, but as you pointed out, mice lack many of the human-specific mutations in HAR-123 to begin with. So how confident are you that the knockout phenotype reflects what the human-specific changes to HAR-123 actually do?
I guess the short answer would be that we're not confident at all. Basically, one of our questions was, what does HAR123 do? And I spent a lot of my talk talking about that. And that was one of our main focuses. And in vivo, we have, as you said, evidence that it's involved in cognitive flexibility. But of course, we only know that in a mouse. And so we don't know if that can relate to humans, but we're obviously excited about the possibility that it might. By the way, of course, our big interest is what's the difference between humans and chimps? And by the way, there's actually evidence that chimps have more cognitive flexibility than humans, actually. I would say the evidence is limited, but there's some evidence for that. So I think it's an intriguing question for the future to try to, or my guess is it depends on the kind of cognitive flexibility, whether humans are better than chimps. So we'll be fascinated to try to deal with that in the future. But obviously, it's a little bit difficult to answer that question in humans in vivo.
Genevieve, I might take on this question at the top for speakers studying speech areas. How does symmetry differences, dyslexic humans with no other learning issues affect your understanding of the necessity of asymmetry? I love the question. Um, I think there has not been enough research and study of individuals with dyslexia emphasizing the enormous strengths that they have and things that make them unique and different in very positive ways to others. Well, one paper on this from Virginia Sturm looked at a group of dyslexic children and found better spatial navigation, so visual memory, and and I think this is good, but it's complex, as increased emotional sensitivity in her neurophysiology lab to stimuli that were potentially emotion evoking, and so we know often that in individuals with dyslexia, there are, smaller white matter tracts in the left hemisphere. And, what happens to the other hemisphere? And is it possible that, and I'm sure it is, that many people with this asymmetry develop extraordinary capabilities, using the other side of the brain?
There's a follow-up question about HAR123 that was in the Q&A. Is there anything known regarding Homo sapiens differences versus Neanderthal and Denisovan?
No sequence differences that we've seen. So the alterations seem to be prior to that.
And then one sent to me for Allison. How does the Nova1 protein sequence in non-human primates compare to modern and archaic humans? Have you tested organoids derived from non-human primate iPSCs and investigated their maturation patterns?
Yes, we've done this before with chimps and other non-human primates. They all cluster together, so humans is really the outlier. We are the only ones with this modern version of Nova 1, and probably part of that is causing our developmental delay compared to other species. So all other primates would have the archaic version of Nova 1.
Okay, next question's for Alex Pollan. Thanks for a great talk. I'm wondering if you view the differential in brain evolution across species in reward areas as an evolutionary trade-off. For example, we seem to have significantly enhanced connectivity within dopaminergic systems associated with our capacities for sustained attention and sophisticated cognition surrounding reward. However, the same systems closely associated with pathological forms of cognition, such as ADHD and addiction.
Yeah, thank you for that question. I think it's really important to consider evolution in terms of trade-offs, and many of us are interested rightly in the genetic and cellular changes that enhance our cognitive functions, but these come at a cost and can change a fitness optimum, creating new challenges for the brain, and certainly new functional requirements for working memory or reward or other things that increase demand on the dopaminergic system are likely to also potentially confer vulnerabilities. I'm not sure how much of the vulnerabilities of our dopaminergic system reflect the new demands. There's some evidence for increased connectivity, innervation density to certain areas of medial caudate and nucleus accumbens, versus how much of the vulnerability comes from the unequal scaling that occurred during brain expansion, the sheer expansion of the territories to which these project. I think both of those models are worth exploring through a combination of comparative and experimental methods. And it's tough to distinguish. And then, of course, the third force would be the increased lifespan. Maybe not for those neuropsychiatric disorders mentioned in the question, but for the neurodegenerative disorders. These neurons must persist for decades longer in humans, coupled with these increased demands. And I think for all of these traits, it's worth considering the trade-offs. And what we're interested in is the secondary idea that the cell types themselves have evolved to our large brain cellular ecology and these new demands, and that we might be able to discover compensatory adaptations that might shed light on protective factors that we could harness for therapeutic purposes. So yeah, thank you for that question.
The next question from the audience is for James Rilling. What do you think drove the evolution of these neuroanatomical differences? Did a need for language come first? Was it the chicken or the egg?
Yeah, well, hi everybody. It's great to be here. Yeah, it's interesting to think about why language evolved in only humans among all the primates. And there's some different ideas about that. One idea that I think is compelling is that language may have evolved to allow us to transmit culture, especially, across generations, that we accumulate this body of knowledge and somehow we need to transmit that to the next generation, and language facilitates that effort. Language, the other really cool thing about language, very powerful thing about language, is that it allows us to have knowledge about things that we never directly experience ourselves, right? So think about how powerful that is, that something can happen somewhere else in my environment and I will not have witnessed it, but someone else who did can tell me about it, and it expands my knowledge of my environment dramatically. So that's something else that I think we should think about when thinking about the evolution of language. And then also the ability to just kind of have this sort of shared intentionality with others, so that we can collaborate on things. I think all of those are potential selection pressures. So anyway, those are some of my thoughts on that interesting question. I'm not sure that I fully answered it, but yeah.
These are hard questions to answer, I think. So that was a great job. The next question's open to everyone. So we'll see who wants to answer first, and everyone can take a shot at it if they want. How do we escape the accusation of human exceptionalism after hearing all these amazing examples of uniquely human brain specializations? Go ahead, Alex.
I think I could take a stab at this from a few different perspectives, and that is a great question. It's one of the reasons I always like to use the word specialization instead of unique adaptation. Different lineages across the phylogeny are specialized for their own environments and optimized in a way that we would not be. I think one way we can kind of tackle this and take the caution suggested by the questioner, at least from a genomic perspective, is to look equally for signals on the human lineage as well as the chimpanzee lineage or other lineages. And we heard the beautiful talk about HARS. And, if there were more time, folks who have identified human accelerated regions have also reciprocally identified chimpanzee accelerated regions. And now with the Zoonomia Consortium, we can do this across any branch. And that allows us to ask whether certain functional ontologies of genes might be uniquely changed in human versus recurrently changed in other lineages. And it may give us perspective on what is truly distinct on our branch. And so I'll open up the discussion there, but I think this is absolutely worth considering. So thank you.
Anyone else?
I think Alex has done an extremely eloquent job explaining this. But the reason we are in this situation that we can do this, we can think about it. So it's not that we are not recognizing other species. The second one, so it's part of our curiosity and our ability to do this as a species. So that's why there is a bias. But there is a second bias, which Alex and others have already mentioned. We are interested in human biology and diseases, and there is evidence, and as my colleagues have done extremely well, is that, that some of the susceptibility may come from, unique features and changes across the human lineage. So we need to also explore that. To what extent that explains, that's a separate question. So those are two reasons why we have this extreme anthropocentric bias in explaining this, just because we are the only one that can do this, or at least study this. And secondly, we think that this is important for human biology and diseases and conditions.
And there's an argument that we could learn from convergent changes in other lineages. I agree with the point. There is maybe rightly some human exceptionalism because we are the species that's able to study and ask these questions. That said, our brains only reached the size they did in the last few million years. And other lineages like the cetacean clade invented large brains 30 or 40 million years earlier. They may not have the same degree of association cortex as Nanad talked about, so they may not have some of our unique capacities for abstraction. But from a cell's perspective at dealing with the challenges of connectivity in a large brain, they may have had an order of magnitude more time to adapt to the challenges of connectivity in a large brain environment. So we may be able to learn cellular adaptations by moving beyond the human exceptionalism perspective.
I might give a slightly broad answer to the question. Thank you, Alex. Really well said. I think we have to understand our deficiencies as a species as well. And, we have wreaked unbelievable havoc on this planet, destroyed, species after species. So I think understanding why that happens, what are the deficiencies in the human brain that allows this to continue and, and how to remedy it is, I think, a pretty important question for the survival of the planet.
Well, the next question is specifically directed to Alex. Is there evidence that the connectivity issues you highlighted were indeed the selective pressure for the dampened response to oxidative stress we see in the modern human brain? Or is it possible that exposures to pollutants, such as fire smoke or the lead exposures discussed by Allison, could have driven that change?
Thank you. That's a great question. And sometimes it's easier to approach the how than the why. And when we want to explain the why, we can come up with potential stories, but these could be considered hand waving. We started with a hypothesis that there might be compensatory adaptations that buffer increased metabolic stress, and we found some evidence consistent with that. I think to defend whether these are adaptations would take much stronger evidence. It would require both functional evidence as well as evidence for signatures of selection on the genomic level. But then your question is even more challenging, is what drove these adaptive signatures? And we have a plausible hypothesis, but it would be very difficult to fully address that here. We could think about what is most likely based on when these are manifest and which cell types. Do we see it broadly across tissues in the body where you might be managing, say, if the hypothesis is pollutants, do we see things in the liver or the kidney or other changes that have kind of co-evolved or adapted with this? Or is it very specific to a vulnerable population of neurons that would be more affected by unequal scaling or new functional requirements that might be more consistent with this connectivity vulnerability hypothesis? So answering the why is tough, but it is something that I think drove all of our interests. So thank you for that.
Yeah, why is hard. Okay, the next question is for Joe Paradiso. In your talk, you mentioned that we now have true AI. Were you referring to artificial general intelligence or to what Dario Amodei calls powerful AI in his essay, Machines of Loving Grace? Do you think it is important to maintain distinctions in AI terminology?
I wouldn't say, I mean, this is a slippery slope because eventually one leads to the other. This is powerful AI compared to what we had before. Is it a transition or is it a rapid evolution? But between 2020 and 2023, suddenly the natural language problem was solved. This is something we worked on forever. LLMs just nailed it, and we were all kind of shocked that they did it so early. Again, I'm speaking to a group of experts, but the synaptic density of the equivalent neurons was like 10% of the brain, and suddenly you had this thing could speak multiple languages and know all the stuff that would make mistakes, but it was incredible what we had achieved when no one expected it to wake up like that, so to speak. So yeah, we crossed the threshold, and that's what I was referring to, right? Compared to before, everything was fragile. We'd make these models and they would kind of do some things, but they wouldn't deal with anything that was out of context. It got to a point where a lot of these things were solved. And of course, who knows where it's going to end? I mean, there's so many scenarios. I gave a few. We have to see. Things will happen fast, I suspect. But I'd say right now, powerful. General AI, what will that be? What does intelligence really mean? These are deep questions that people in the room have been dealing with biologically for years. Now we have artificial brains, so to speak. We can begin to study and maybe answer some of these philosophical questions as well.
Okay, the next question is for Miles. Is there any indication or speculation on, here's another why HARs are so small?
Interesting question. I suspect that'd be a better question for Katie Pollard, who was the first person to kind of start this field, and others that have used bioinformatics to define them. I think it mostly comes down to bioinformatics and that short sequences are easier to identify that have undergone these statistically unique changes in human, but not elsewhere. I don't think that they're necessarily the functional units. The functional units could be smaller than HARs, and they could also be bigger than HARs. So I think most likely it's a biostatistics reason for their size.
Great, thank you. The next question is for Dean. The questioner asks, I'd love to hear more about the hypothesis that motherese came before full language. What are the differences between the two that makes you think this motherese infant-directed speech is common, but not a cultural universal in humans? Does this pose a problem for the theory?
To begin with, I've addressed the allegation or the assertion that motherese is not universal by looking at those specific societies that were claimed not to have it. And it turned out they do have motherese. It's just different in different cultures. For instance, if you have a culture in which eye contact is taboo, you don't have eye contact in the motherese. What was the beginning of the question?
Yes, sorry.
Oh,
The hypothesis that motherese came before full language.
okay. Only humans have motherese to the extent they do. Lots of animals have contact calls and a little bit of interaction between mothers and infants, but only humans do. It's an extremely special register in which parents, it can be the fathers too, or other individuals, speak to babies. It is shown, there are lots of articles that shown it absolutely bootstraps acquisition of language in young infants. They're born without language. They're exposed incessantly to motherese. And by around about the end of the first year, they're starting to get language. And part of the reason is because one of the functions of motherese is that it highlights certain things infants need to know in order to acquire a language, like how to parse a sentence. People speaking motherese, most are completely unconscious of the fact that they are hyperarticulating vowels, for instance. So there's a large literature which shows that motherese, the more motherese you speak to your infant, the better in terms of its acquisition of language. So don't worry about, are you speaking down to your infant? They like it. So it's unique to humans. In humans, it precedes and bootstraps the acquisition of language. So it seems logical then to ask, well, when did it arise? Other animals don't have it. When in hominid evolution did it arise and how? And that's what the putting the baby down hypothesis addressed. And I've now corrected it in light of David Lindsay's work. I would now call it the picking the baby up hypothesis because of some very important caveats he raised. So yeah, I think Mother E's was, it evolved. It evolved into our lineage, and we know its function besides bonding, expressing love, and becoming educational when the infant is old enough. Because in Mother E's expands as the infant develops, the Mother E's changes to accommodate the infant. I hope that addressed most of the question.
Yeah, that was great. Thanks.
Thank you.
Okay, the next question's for Allison. Do you have any thoughts on why, again, the modern Nova 1 version only evolved in humans as a response to lead, if, as you mentioned, lead has been contaminating humans and other primates for about 2 million years? Why was this change specifically selected for in humans, but not our relatives?
Yeah, that's a great question, and I don't have the answer. So I can only imagine that this is all about genetics. So it turns out that the mutation arises in modern humans and then was selected. I mean, I would say that this could have happened in other species as well. It just happened to us, and we have the right environment, the right brain, the right cell, the right genome for these to be selected, especially on this cluster with FOXG1. I think this was like an important observation that we have those as well, and we know that these regulation of those genes might be different in other species. So I think we were just the lucky ones.
Well, can I jump in here? Uh, Perhaps it's kind of a bad news, good news situation where, and this is obviously highly speculative, but what if the human precursors were in a particularly lead rich area? And of course, at first that was bad news. But the good news is then there was selection for this changes in Nova, which then led to good news, which is, good things for the brain. We became smarter. I'm obviously simplifying it, but you know what I'm saying? That sometimes a bad situation can lead to a good situation.
Yes, we hope all bad situations lead to good situations. Great. The next question's for Nanad. The question is, the effects on neuronal development trajectories in knockout mice brains in these studies is intriguing. Have any behavioral studies been conducted in SATB2, CYP26B1, or mice2 knockout mice? And what do these animals look like behaviorally?
So very good question. And the answer is yes. The behavioral studies have been done, and they did poorly, at least in case of maze 2, in tests that require working memory and prefrontal cortex. But you have to, again, I think Miles said that really clearly, nicely, and eloquently. It's very hard to study behavior in mice. We are separated by hundreds of millions of years from last common ancestor. So I think whatever you get there, you have to be very careful with what you are trying to interpret in the context of human evolution and human cognition and or behavior. So just to let you, also one important thing to also understand, these are extremely important genes. And so, we shared more than we are different, even with other non-human primates. So we have to keep that in mind. And also in what Miles showed was very beautiful example of a HAR that should have and has human-specific properties, but yet it is affecting a gene that is probably highly conserved. And that gene is also affected by lineage-specific events in other species, including, let's say, in chimpanzee lineage. So again, we have to, we are really peeling an onion here, and we are maybe a couple of layers, maybe even one layer. And so we have to be very careful. Yes, there are clear behavioral deficits in all these mice. The phenotypes are very profound, which also tells us that probably the genes that are under some kind of a human or primate specific selections it's good to have a gene that is being changed in its expression levels and or spatiotemporal that are important developmentally, genes that are important. So that's all great. But I have to just say that yes, there are behavioral deficits, just interpretation of them has to be done with a lot of caution.
Yes, totally agree. Thank you. The next question's for Bruce. In your patients with degenerative neurological conditions, did you observe upregulation in positive emotion moods associated with proclivity for artistic endeavors, or was there more or less a neutral affect present?
Yeah, it's a great question, and I don't think we've answered that in the subset with artistic creativity. The person who studies this in most detail is Virginia Sturm, and she's contrasted frontotemporal dementia, which hits frontal circuits, salience network, sometimes with a upregulation of posterior brain, versus Alzheimer's, where there's a loss of default mode network, posterior brain circuits, and sometimes upregulation of the salience network. And so in the Alzheimer patients that she studied, who are amyloid positive but not symptomatic, there are some emotional signals. There's and I think also in one study there have of a cohort, the Baltimore longitudinal aging study, there are personality changes that seem to precede any memory cognitive issues, and I think they are associated with things like increased neuroticism, increased anxiety. And more introversion. So I think these diseases affect emotions very differently. And then there's the left versus right, which I think also influences exactly how we see these emotional disorders. But I think it's an astoundingly good question. What is happening to the emotional system in these artists, and we just haven't even touched that.
The next question's for me. I feel like almost Alex could answer this a little too. What is the potential downside in having a reduced number of mature oligodendrocytes in humans? What's the trade-off there? So I kind of alluded to this a bit at the end. I mean, of course, we don't know, but we speculate that this may be linked to human risk for demyelinating disorders like multiple sclerosis, but we don't really know how this change in ratio affects differentiation or if there's increased apoptosis of one versus the other. So this is something that we're trying to study in the dish. But right now we're going after really the disease aspect of it. Alex, do you want to comment?
Yeah, I think it's a great question. I always love to think about trade-offs. I think we could extend this from the oligo lineage alone to thinking about trade-offs reflecting the protracted maturation time in humans. We have this potentially unique period of early childhood. We have extended adolescence window. Our development is slower in many capacities, and one of the trade-offs related to protracted development, including kind of delayed maturation of oligodendrocytes, relates to these increased windows of vulnerability to environmental factors. We have more plasticity in our circuits, but that may also, in a general way, confer vulnerability to neuropsychiatric disorders. This additional plasticity in wiring could lead to kind of aberrant reinforcement of pathological circuits. And so I think those may be some of the most dramatic trade-offs related to this slow maturation that we face. And Nanad has done extensive work on the molecular staging of these periods across species. So maybe I'll turn it over to you.
Thank you. Thank you, Alex. And there is very little to add to this. I mean, I accept that I do completely agree, and Alex has done a very good job trying to describe all of this. There is a reaction to every action, and so whichever cell type you change, whatever you change in the brain development, there's something else you have to pay a price for it. And this is true for all species. It's like if you look at marsupials, they are born prematurely, and so they develop a specializations to really deal with that, okay? And so, they also have some advantages and many disadvantages to their life history. Uh, we have to also be careful to what extent changes in human life history, not just but including aging and stuff like that, are due to our environment and lifestyles. We all treat each other differently than, we are caged animals or living in the wild, as Bruce says, in some cases badly. Uh, so I think those are all things that we have to take in consideration when we're looking at the changes in life history and pros and cons of every change. Nothing is free or without consequence.
Yeah. Excellent. Thanks, team. The next question's for James. There are a number of species, such as dogs, that seem to outperform great apes on cooperative communication and sometimes theory of mind tasks. Based on your research on the default mode network, would you expect convergent evolution of a novel PCC to MPFC pathway, or the emergence of a distinct neural mechanism underlying these abilities?
Yeah, what an interesting question. I guess because, humans have been living with dogs for such a long period of time and working together with them and selecting them in different ways, it makes sense that dogs have come to understand humans better than maybe other species, and that and that's part of why they're great companions, is because it seems as though they have some like human-like social cognitive abilities. And so, yeah, what an intriguing question. So, do we think they have a default mode network, or they have uh, specializations related to theory of mind? I'm not sure. There are different levels of theory of mind, and so sometimes when we're talking about theory of mind, we're talking about really high level, like, you know, do you know what I know? Can we both share the same goals and ideas? And sometimes it's lower level, like, do I understand your intentions? And that sort of thing. And so it's hard to know exactly. I don't know enough about dog cognition to know like what level of the theory of mind hierarchy they're at. But yeah, that's an intriguing question. There are some people who are doing a little bit of dog fMRI. One of my colleagues here at Emory, Greg Burns, has done some, and I think it's a really exciting area for exactly this reason, that they have so many unique or special sort of social cognitive abilities. But yeah, thanks for a great question.
That's really fascinating. Dog fMRI studies. That's great.
Yeah.
Okay, the next question's open to everyone. What do the speakers think newer methods such as spatial transcriptomics might add to comparative brain studies beyond what we've already learned from bulk and single cell approaches? I mean, I think it's the future of genomics. I'm a single cell genomics person because we can look for rare cells, we can look developmentally where cells are migrating in the tissue. We can get a larger area. When we dissociate the tissue, we lose so much information. But I've already drank the Kool-Aid, so I don't know if anyone else on the panel has other thoughts about this.
I'll just say I totally agree.
Yeah. Alex, yeah.
Yeah, so we know so much about the genome sequence differences between species. And of course, there's this gap between genotype and phenotype. And, Allison talked about how we can take a strong effect, potentially candidate mutation from signatures of selection, and then try to link it to phenotypes by reconstructing it. We can tell some genomic regions that are under adaptive selection, but it's very hard to tell with these neural cell type or neighborhood phenotypes what is selection and what is constrained. And I imagine by really extending our comparative phenotyping to the level of cellular organization, we can get a sense for what kind of neural circuit and cell type motifs are constrained within species but divergent across species. And we might be able to get more of a signature consistent with adaptive selection for some aspects of nervous system organization. And, the big challenge, of course, is the limited amount of tissue that we can work with. And there are resources out there that are ethically collected, but it's difficult to use these. It's also a challenge to think about when is the right time to use these scarce resources. When is the technology ready, given that we might not be able to continue to expand these collections of postmortem samples. So I think there's things to think about. I'm certainly enthusiastic for more extensive comparative phenotyping, including spatial transcriptomics, as well as some of these diffusion tensor imaging and MRI approaches that we heard about.
If I may add to this is I think, an excellent question, but also it's a future, as you said, Jenna, and Alex has also really pointed out to what is important. But one thing that we need to do better is to integrate across subdisciplines and scales and modalities. And there is a lot of classical literature that is really important in neuroanatomy. But we have a single cell where you are just breaking up things. So there'll be a lot of literature in imaging. And so I think the spatial technologies, whether it's transcriptomics or something else, will, I think, help us bridge that. And I think this is particularly also important for development, where things are extremely dynamic. There are transient cell types, there are transient circuits that we know from working experimental animals. We know a little bit about it in human brain development, but we actually have very little knowledge. I mean, almost none in what happening in closely related species, particularly chimpanzee. So for that reason, I think it's absolutely something that I think will be critical for moving field forward.
The next question is for Allison. This is a long one. What do you think are the best and worst case scenarios as archaeolization technology continues to develop? Do you have any thoughts about a recent paper calling for regulation in this space, pointing out that wealthy donors could theoretically fund private efforts in the extinct human lineage in the same way Colossal Biosciences has been pursuing the dire wolf, woolly mammoth, and other so-called resurrections?
Yeah, that's a great question. Yeah, yeah. There are thousands of differences between humans and the Neanderthals. When I say a 6 to 1 variants, these are protein coding genes. So even if we change the 6 to 1, there's other thousands of regulatory regions that would be very hard to manipulate genetically, at least with the tools that we currently have to reconstruct entirely a Neanderthal genome. Someone might even think about reconstructing using like haplotypes from humans, but even though, as far as I can tell, will never be like a complete Neanderthal. So I'm not too worried about that. But there is a cool movie called William from Team Disney, who discussed this possibility, of course, not through genome editing, but supposedly that you can find an intact cell from a Neanderthal that is frozen and you revive the nuclear and do like a nuclear cell transfer into a human egg, and you might raise a Neanderthal. But even though it's on the fiction level.
Yes, thank goodness, still fiction.
Oh, and we should touch on that there are like legal prohibitions against germline editing. I don't know how they would apply to somatic cell nuclear transfer from your frozen tissue, but there's many legal and ethical restrictions on editing the human germline that govern this. I think what Allison does in vitro in cell lines, testing a small number of these mutations at once for experimental studies, is very distinct from this concept of trying to create a human organism carrying many Neanderthal variants. Um, but I think an important addition is that many of these variants still exist today. Like 30 to 50% of the Neanderthal genome is preserved among humans, just at low frequency in any individual human. So for many of the variants, but not the 61 that Allison mentioned, we actually have the opportunity to study and learn from their function by standard genetic association mapping studies among humans.
Okay, the next question actually is for you, Alex. In the common garden environments where you grow cells from different species together, how do you rule out potentially unwanted or unnatural signaling that could change cellular behavior?
Um, yeah, this is a great question. It's a pretty new approach to mix cells from different species in the same environment. And also, anytime you mix human and non-human material, there's ethical considerations, which we've considered extensively before these studies. But in terms of the experimental points alluded to by the question, in our studies, we both do within species pools as well as between species pools. And one of the first things we analyzed was to what extent is gene expression divergence governed by extrinsic mechanisms, like the question asks, versus cell intrinsic mechanisms. And some of the most exciting candidate genes from recent studies could point to either mechanism. Like Deborah Silver's lab has this example of a HAR near the gene frizzled 8 that could increase Wnt signaling in a cell intrinsic way. But then Sophie Salama, David Hausler, Pierre Vanderhagen, and others have found a human-specific gene, Notch2NL, that could increase Notch signaling in a cell extrinsic way. And so what does it look like when we do the within species versus between species organoids? And what we find in our hands, at least, is that nearly all of the cell type specific gene expression divergence is governed by intrinsic mechanisms. And that supports the use of these mixed species organoids for discovery of intrinsic mechanisms. That's not to say that there aren't extrinsic mechanisms in the right cellular environment, and we're very interested in investigating this as well. But there's a lot of evolutionary theory that modular cis-regulatory changes driving gene regulatory networks will guide adaptive changes because they're favored under selection, and those would act cell intrinsically. And so we actually think it's an advantage of the system that we can dissect what's intrinsic and extrinsic. And then the other part is that there's a practical utility to mixing these cells, and that controls for batch effects allows us to scale to many individuals to identify species differences rather than individual differences. And then for our experiments where we want to deliver some kind of external environmental stimulus, in our study, we delivered an oxidative stress stimulation. We can really control for these being in a similar cellular stress oxidative environment going into the experiment. And so that's why we made these experimental choices. I would love to discover what the small number of extrinsic changes are, because that could point to potential driver changes like this Notch2NL example. But we can also use this system to discover extrinsic changes, and that's something that we're interested in working on moving forwards.
Thank you. The next question is for James. The gradation of size of the lateral temporal cortex was intriguing, as well as the finding of pronounced difference between this area in humans and its chimpanzee homologue. I wonder about cell types there and if there are mirror neurons or connections from mirror neurons, for example, from the IPL present in the area.
Yeah, so the pathway that we were focusing on, the arcuate fasciculus, is really connecting the temporal lobe, so the posterior superior temporal gyrus, the superior temporal sulcus, and the middle temporal gyrus, as well as the inferior temporal gyrus, so lateral temporal cortex, with uh, the frontal lobe, with prefrontal cortex, and especially the ventral prefrontal areas like Broca's area. So that pathway doesn't include the parietal lobe. And so where there are mirror neurons, but of course, we know there are mirror neurons in Broca's area, which is part of the pathway. And so I think, yeah, it's interesting to think about differences in those mirror neurons across species. And so I know there's been some work done on that. Of course, macaque monkeys have mirror neurons in the homologue of Broca's area. And I know human mirror neurons have some properties that you don't find in macaques. And for example, they respond to intransitive grasping movements, whereas macaque mirror neurons don't. So yeah, I think that's it's important to think about. We're working at more of a systems level, macro level, but it's important to think about underlying cellular differences as well.
Okay, the next one's for Bruce. Was there any indication of correlation with experiences of mystical type encounters, like notions of the divine or supernatural that accompanied the conditions?
Yeah, the one subgroup of patients that we see this in, and is a group. Teresa Wu wrote the first paper on this. Are patients who degenerate the right anterior temporal lobe, and we see increased religiosity in this group. We tend to see also hypergraphia in this group, but the religious intensification, philosophical intensification is a feature of this subgroup of patients with frontotemporal dementia. So there's something about the balance between the right anterior temporal lobe and the left that I think releases this phenomena.
Interesting. Thank you. The next question's for Nanad. When we talk about information flowing through cortical circuits, signals going up from sensory areas versus predictions coming back down from association cortex, how precisely can we actually track or decode that right now?
Oh, thank you. It's a wonderful question that I've been recently reading more extensively, but I cannot answer that question. I just don't have the knowledge and expertise to answer that question. So maybe somebody else on the panel. Those are very technical details that just I don't have that knowledge to understand in the imaging. So I apologize. It's a fabulous question. I just don't have the expertise.
Hard question to answer. There's one more for me. Do I believe that the between species difference in immature oligodendrocyte markers persists throughout the lifespan of humans, or expected to follow a degenerative course across all three species you tested? Yeah, that's a great question. And that's something we're trying to answer by sampling across the lifespan with a different project from birth through adult. And for degeneration, I mean, that's a separate kind of question on top of that. One is developmental, and then one is degeneration. So we were at middle age when we did that study, and we need to go before, we need to go after. But yeah, I really like that question. I'm going to do a wrap up, I think, unless anyone has any follow-up answers to any of the questions that were asked already. Okay, so to our speakers and to everyone still in the audience, this is the end of the question and answer period. So I'm just going to take a few minutes now to kind of do my best to summarize the day. It's a lot of information. But first, I really want to thank all of the speakers. These were amazing talks. I'd also like to thank all of the symposium participants who I can't see for listening and asking all of these really excellent questions that have, I think, really got all of us thinking further about our own research in the future. So today we've explored many different aspects of the human brain, what we call the idea organ, and the role of evolution in its function at many different levels, really. We've discussed how the brain has biologically evolved, how it's culturally embedded, and what I think is really cool, how technology has allowed us to extend that beyond our own intrinsic biology. So at the biological level, we've heard many different things. We've heard about developmental mechanisms that impact circuits and how these shape neural architecture. And we can measure that neural architecture in a number of ways through brain imaging and through model systems. And then how ultimately does this neural architecture underlie human behaviors like tool use or picking up or dropping babies to the emergence of language and theory of mind? And then how did this tool use, language, and social cognition enable culture? Beyond behaviors, we heard about cell types and alterations in cell types, whether it's a specific cell type, such as the von Economo neuron, or proportions of cell types like oligodendrocytes. How do genes influence these features? For example, how do HARs influence cell differentiation or proportions? Or the use of genes from understanding ancient genomes, such as those from Neanderthals, to identify modern human-specific alterations. And then taking those genetic alterations and circling back to behaviors like language. So we saw the case of NOVA1 intersecting with FOXP2, a so-called speech and language gene, or how our understanding of cortical development that's important for brain evolution in the case of mice too. So beyond then cell types and genes, we also talked a lot about modeling these features to dissect them and to understand them and really get some experimentally tractable information. So we can do this by doing evolution in a dish or in a model system. We heard a lot about cells, organoids, and model systems like rodents. We also, what was kind of refreshing to me and something that I don't often get to hear about in a similar symposia, the intersection of the human brain with art, both visual and musical. Music came up in the talks in several different ways. We also talked a little bit about pathology, what happens when the brain goes bad, but then refreshingly how that can reveal human-specific functions, like in the case of FTD, revealing new creative abilities. And then finally, how do computers intersect with our biological systems to allow our brains to reach full potential? Again, hearing music and computers in the brain intersect, and the future of augmentation, whether that's to play music or to even change one's surroundings, is very remarkable. And I'm just going to steal one of the quotes from the last talk, from Joe's talk, where he said, "What will the human become as everything merges?" And so I think the future directions are really exciting. We have some limitations. We kind of touched upon them a little bit in the Q&A with respect to ethics and what we can actually manipulate either in the model systems or even in the human brain. We can't really change our genetics, our circuits, or our cells ethically or even with the tools at hand. But we can clearly model some of these features. And now we've seen that we can augment them in living humans, and then we can study behaviors and then how those new behaviors with augmentation may impact culture. So we're kind of affecting our own evolution in a sense. So there's so many more discoveries to make about the idea organ. I mean, this has been a remarkable day. Again, thanks to all the speakers, and I'm going to pass the baton to Pascal for final closing remarks.
Thank you so much, Geneviève and Alison, our two co-chairs, for this amazing symposium, and all the speakers. And I will thank all of you for attending online. This was virtual. If you want to catch some of the talks you missed or any of the hundreds of past CARTA talks, you can do so on our website or on YouTube. We also want to let you know that we will have an in-person symposium in fall that will be about we are what we ate, the diets that fueled human evolution. There will be food involved. So we look forward to seeing you then. And of course, count on your continued support to make these free symposia possible. Thank you all very much.